No. No. Oh, that's the pointy thing.
Oh, is that the tongue?
The pointy things. What do we call those pointy things?
So the collar.
Yes, filiform papillae. Remember they're the one of the papillae that do not bear taste buds. All right? And you can feel that they can be very horny, as in you've ever had a domestic cat lick you. Imagine the filiform papillae on a 450-pound adult male lion, strong enough to lick the fur right off their prey. Okay? Here we can see taste buds. We can see the von Ebner's gland, and this would be what? The duct to the von Ebner's gland, right? So you've got the gland here out of the planus section, but you can see how it empties nicely into the furrow. We've got lots of taste buds here. This slide is labeled a circumvallate papilla, but I expect it's actually a foliate papilla because circumvallate papilla don't have taste buds on the walls of the trench. Don't worry about that so much. This is exquisite right here. You can almost see the taste port, which is slightly out of the planus section, which bears the little microvilli that has the taste receptors on them. If I remember right, the circumvallate papilla mostly sense bitter. Okay. What are we looking at? Low power, I admit. All right, but at this point in the class, you should be able to. What kind of epithelium is that? It's clearly not simple, right?
Columnar.
So stratified, and you want to be thinking until you can go to higher power, probably stratified squamous. All right. This is a nice cross section of the esophagus. All right. Even though it's not complete, I know it's a cross section because I can see that these muscle fibers here, smooth muscle fibers, which are rather large and are what part? Well, what are we going to call those muscle fibers? They're a part of the mucosa. Exactly, muscularis mucosa. The thing about the esophagus is the muscularis mucosa is a lot thicker than you see elsewhere in the stomach, small and large intestine, and the smooth muscle fibers run parallel to the long axis of the esophagus. So if they're cutting cross-section, the esophagus is cutting cross-section. All right? Just to preview what's going to happen week after next when Nigel does the female reproductive system, the vagina has a stratified squamous, non-keratinized. It looks similar to what we're seeing here in the esophagus, although the vagina does not have a muscularis mucosa. So it's worth holding that in your head. So in reality then, the lamina propria is going to be this space right in here. So that the mucosa consists of the epithelium, the lamina propria, and the muscularis mucosa, which makes this space right here submucosa, and we can see nicely these esophageal glands hanging out in the submucosa. This would be the muscularis externa or propria, and adventitia, the only connecting tissue you're seeing is up here. It's been pretty much cut away right here. So here's a nice shot again of the esophagus. Is this a longitudinal section or a cross section? I'm deaf, so sing it out. Exactly, because again, look at the muscularis mucosa. It's cut in long section. So, lamina propria, submucosa, and then the muscularis externa. This shot was taken from the muscularis externa or propria in the mid-esophageal region. All right? So we can clearly see what kind of muscle here? And then smooth muscle. So there's a mixture. So smooth muscle up top, and a little bit right here, and this 1, 2, 3, 4 striated muscle fibers right there. This is a scanning electron micrograph looking at the surface of the stomach. Rather high power. But what you can see nicely are the apical regions of these surface mucus cells, as well as the gastric pit, which is going to go down and form the gastric gland. So this next scanning electron micrograph is just exquisite, and I don't know how they really did it. It peeled away all the lamina propria, which surrounds the gastric pit and gastric glands. So the gastric glands are tubular, and there's anywhere from 2 to 3 per gastric pit that opens up. So the asterisk right here is in that neck region. So this would be gastric pit. The neck, the isthmus is kind of up in here, which is noteworthy because it's the stem cell niche. It's where stem cells that produce the surface mucus cells would rise up, and those producing parietal or chief cells would drop down to the base. Interestingly, the surface mucus cells have a lifespan of 4 to 6 days. You get down into parietal cell, it's about 200 days, and the zymogen cells are 90 to something. So, stem cells can differentiate into cells that are very different and have very different lifespans. All right, so let's take this to standard light histology, and we can see the lumen here. We can see a little bit of mucus left on the surface here. Here we can see nicely coming down into the pit. So imagine that scanning EM I showed you looking down. We were looking down into these pits right here. What do you think this crimson color? You see, if you look carefully, you see them all over. Blood. They're blood vessels. You need to look at higher power. I'm just pointing out something that's obvious. It's a real nice crimson color. All right. Now the blood vessels are in the lamina propria, which closely invest these tubular glands that are sticking down in it. All right. And you can see nicely here, what is this structure? Muscularis mucosa. And again, it's down to the thinner region that muscularis mucosa that we're used to, unlike what we see in the esophagus. Now, if you look carefully at this, you can see that there are 3 kind of layers, color layers. There's this layer that has the gastric pits, and the interface is right here. There's this lighter pinker layer right in here, and then there's a more basophilic layer. So this tri-layered or tricolored tells us that down here we've got more chief cells than parietal cells, more parietal cells than chief cells, and we don't have either up here because we've got surface mucus cells. All right? That's very helpful in identity at a low power, and in reality, if you have a good slide, you can see this by eye. You can see this 3-color layer by eye, but clearly see it if you go to 4x. So that's going to tell you that you're looking at gastric mucosa. Now realize that in the cardiac and the pyloric regions, the gastric glands, for the most part, lack parietal and chief cells. They just have more mucus cells. So same tubular glands, but they're mucus cells. And because they're mucus cells, they're not going to stain. All right? They're going to be in absence of stain, at least in the standard H&E preparation. So, same gastric mucosa, slightly higher power. So this is that interface between the pit and the gland. You can see that we're getting down into the parietal cell layer here. The other thing that is really important here is for you to understand the difference and have in your head the difference between a surface mucus cell and a goblet cell. Now they both produce mucus, no problem with that. But the surface mucus cell is a simple columnar cell next to the same sort of thing. I mean, you can see that by looking at these puppies lined up one after another. Usually the nuclei are at the bottom of the cell. A goblet cell, you remember, has that wine glass shaped curvy thing, and it's kind of a stem. Completely different. If you see goblet cells, you know you're not in the stomach, okay? Vice versa, if you see surface mucus cells, you know you're not in the small intestine, or large intestine for that matter. So it's slightly higher power, this middle layer. There's a pink fried egg. There's another one here. These are all chief cells down here. Now notice, because these the gland part don't just stick straight down, they kind of curve around one another. You rarely cut into the lumen of the gastric gland. But right here we can see it reaches. So that's the luminal area, and then we lose it in the plane of section. There's the lumen. Here we're just raising the outer surface of that tubular gland. Now, this is a classic textbook. I think this image is even in MedPix that most of you didn't get to see today. All right. First thing is, if you follow the gastric mucosa, here we've got these pits that are invagination, so they're in from a surface, a little bit of surface mucus here. Coming around here, it's folded, so we've cut these pits in cross section. But there's a big difference between histology here and what you're seeing here. You see nicely villi, a little too low power. You might even be able to pick out the occasional goblet cell. The other thing that's really important to get a feel for is the muscularis mucosa coming right like that. What's that? Brutus gland, exactly. And here's that pyloric sphincter, which is an elaboration of the intercircular smooth muscle at that point. So we've gone from a mucosa that is all invaginations in the form of gastric pits to a mucosa that has evaginations, fingers pointing out, as well as invaginations, the intestinal glands going this way. This is a quite nice scanning electron micrograph. We're looking down on the duodenum here. Well, actually, this is jejunum. You can see the villi sticking up, and the actual arrowheads point to the openings of these intestinal glands, or we give them another fancy name, crypts of Lieberkühn. All right? Here, we're in the small intestine. What part of the small intestine? Duodenum, and that's because Brunner's gland. What we're looking at here are all intestinal glands. That's lumen up there, so you can see, follow it down, it's an invagination. Notice that the intestinal glands are surrounded by lamina propria, whereas the villi have a core of lamina propria. All right? And if you hallucinate, you might be able to see the slight red staining, which would belong to what kind of cells that are at the base of the intestinal gland? Pannus cells. And here's, this is exquisite right here. You clearly see these guys. They're cranking out lysozymes and defensins, things that help in a protective mode, as Nigel mentioned. Here we can see, what's this structure? Muscularis mucosa. And again, it's really thin compared to the esophagus. And of course, we've got Brunner's gland right there. Nigel showed you this in lecture. So this is the jejunum. See, nice villus here. Again, it's got a core of lamina propria, whereas the intestinal glands are surrounded by lamina propria. And here, nice, that goblet cell. Really distinct from the surface mucus cells of the gastric mucosa. Oh, one thing to mention. The stem cells in the intestine are at the base of the intestinal glands, so they migrate up. Remember, in the stomach, they're at the neck of the glands, and they migrate up and down. This is an immunostain for enteroendocrine cells. I don't know what type, but you can see that they're scattered about the base of the intestinal glands here. What part of the GI tract are we? Exactly. And you know that because of these nice pairs of patches. Even at this low magnification, if you look at these villi, you can see that the lining has, you can see the goblet cells in it. See that, you know, where the mucus is washed out, pale staining. If you look at this villus right here, note that it's been heavily, well, first, muscularis mucosa, you can pick it up, it's been breached there, pick it up, you really have to, you can't even see it because the lymphocytic infiltrate is so intense. And then if you look at the epithelium here, you're not seeing any of the goblet cells that you see up here. That's because the cells are all what are called M cells or microfold cells. I think I put, yes, this is really exquisite. So let me trace out this M cell. It's basically kind of looks like an attenuated horseshoe, all right, sticking down like that. Adjacent to standard enterocytes or simple columnar absorption cells with microvilli. This guy doesn't have microvilli, but folds that allows it to endocytose bacteria, that sort of thing, which basically, because the cell cytoplasm is so attenuated, it can pass it to this pocket underneath that contains lymphocytes, macrophages, so it basically can initiate an immune response, the result of which is these stimulated lymphocytes move to a lymph node underlying in Peyer's patch, stimulate plasma cell formation. Plasma cell starts secreting IgA, which is then exocytosed back out into the lumen where it can complex with whatever antigen it's been made to. All right? That's called immune exclusion. Same sort of thing happens in the salivary glands. A lot of IgA is dumped into the saliva. So this is a nice scanning EM shot, again, of the colon, the surface of the colon. And you can see all these little depressions, which would represent the openings to the intestinal glands. All these little bumpy-like things would represent mucinogen packets, which have not yet hydrated and formed the lubricating mucus, which is so important for the large intestine. So this is a classic shot where lumen, and you can follow, that's one intestinal gland, another. Here we can actually see the lumen, cut to the lumen, a little bit of lumen there. Here's your muscularis mucosa. A lot of your slides showed a section that would cut through here and look down on it. So you get all these little circular profiles of whatever, however deep you cut into the intestinal gland. Nice shot of the appendix, a cross section and a longitudinal section. Even at this magnification, you can see all of the lymphoid nodules, many of which have germinal centers lining the submucosa and mucosa of the appendix. At higher power, you can see that, as Nigel mentioned, the mucosa is colonic, or that comparable to what you see in the colon. Lots of intestinal glands full of goblet cells, right? You can see that you've got the lymphoid nodules beneath. They've moved up from the submucosa, almost up into the mucosa. Different than what you see in the ileum, the lymphoid nodules are completely circumferential. They're completely around the whole mucosa, submucosa, unlike Peyer's patch, which are aggregated just on one side. I realized I forgot. Harry mentioned that there was a bit of difficulty between Paneth cells and Brunner's gland. So let's keep in mind that Paneth cells are individual cells that are part of the intestinal gland, whereas Brunner's gland is a gland that's usually in the submucosa, the duct of which can empty into an intestinal gland, but completely separate. So that'll do it. Next week we've got exocrine glands and endocrine glands. Have a good weekend.